Hello, and welcome to this Microbiology Bio 203 video lecture from Chapter 17 of your OpenStax book, Innate Non-Specific Host Defenses. I'm Mr. Kennedy, and I'll be your guide as we briefly explore this topic together. Learning outcomes from Chapter 17 include being able to describe the lines of defense of the immune system, describe the physical and chemical defenses of the innate immune system, describe the different types of white blood cells and their functions, and explain the processes of inflammation and fever. We begin today by taking a quick look at immunity. Susceptibility is the lack of resistance to a disease, while immunity is your ability to ward a disease off. There are two arms of immunity: innate immunity, which provides defense against any and all incoming pathogens. These are defenses that we are born with, and they are somewhat limited in their effectiveness. Adaptive immunity is sometimes thought of as learned immunity. This is immunity developed over time and geared towards resistance to a specific pathogen. Learned immunity is gained from exposure, through breast milk, or through vaccination. This is an illustration to help you understand the lines of defense that a pathogen must breach in order to successfully invade the body. The first line of defense, the most generic, are your skin, mucous membranes, and antimicrobial substances secreted there. These are defenses that are non-specific. As a second line of defense, we have inflammation, fever, and phagocytosis, also non-specific defenses. As a final line of defense, we have the adaptive immune response operating at the humoral and cellular level, geared towards protection against specific organisms. Physical barriers and defenses preventing entry. The body has a number of physical barriers to protect you from foreign invaders. We begin with cellular barriers, which include your skin and mucosal membranes. The epidermis of the skin is made up of tightly packed cells impregnated with keratin, which is a protective protein. The skin operates as an effective barrier against foreign pathogens. However, it can be torn or breached. Mucous membranes. Mucous membranes line all of the necessary openings to the skin. There, cells produce mucus, and ciliary escalators move that mucus to a place where pathogens and the mucus itself can be processed, such as the digestive system. Mechanical defenses. Mechanical defenses include the shedding of cells or moving liquids. The skin itself sheds cells copiously throughout the course of the day. Those cells are replaced from underneath. Any organism attempting to colonize the skin must deal with the fact that the surface that it is growing on may simply break away as the colony grows. Moving liquids, tears, saliva, and urine can actually wash or remove foreign pathogens from certain areas of the body. Resident microbiota is a final physical barrier to colonization by foreign pathogens. Your resident microbiota live in harmony with you and occupy niches available to these foreign organisms. Their simple presence provides the competition necessary to prevent the foreign pathogen from gaining a foothold on the body. Chemical defenses. Sebum forms a protective film and lowers the pH of the skin to about 3 to 5 on the pH scale and provides an effective chemical defense against foreign pathogens that cannot tolerate such acidic environments. Earwax lines the ear canal and also has a low pH. Lysozyme, found in perspiration, tears, saliva, and other tissue fluids, is an effective chemical defense as it breaks cell walls and microorganisms. Gastric juice and vaginal secretions also protect openings in the body through the reduction of pH in the areas mentioned. This table will provide you with a summary of antimicrobial peptides that are used as defenses by the body. Pause the video and take a moment to familiarize yourself with each antimicrobial peptide, where it is secreted, the pathogens inhibited by it, and the mode of action of that inhibition. Plasma proteins, or acute phase proteins, are listed in this table. As was the case with the previous slide, pause the video and make sure you familiarize yourself with each of these plasma proteins and their mode of action. The complement system. The complement system involves less than 30 serum proteins activated in cascade, similarly to coagulation process of blood clotting. The complement system involving more than 30 proteins affects phagocytes, vasodilation, membrane attack complex, or cytolysis, and opsonization, or immune adherence. The membrane attack complex is formed by C6 to C9 proteins, and its job is to make a hole in the cell membrane, which can lead to the osmotic lysis of the foreign pathogen. Complement activation. There are three scenarios that may play out in complement activation: a classic, alternative, and lecithin pathway. The classic activation system involves antibodies bound to the microbe provoking activation. The alternative is the microbial components directly activate the complement. The lecithin pathway involves certain sugars, lecithins, on microbial cells that activate the complement. Cytokines. Cytokines are soluble proteins. They provide communication signals between cells and include things such as interleukins, chemokines, and interferons. This is an illustration of how cell-to-cell signaling can be accomplished. Inflammation-eliciting mediators. Here we have a table of a few inflammation-eliciting mediators. Histamine, kinins, prostaglandins, and leukotrienes are 4 examples. Each has a specific role to play, such as vasodilation. Take a moment to pause the video and familiarize yourself with each before moving on. White blood cells. White blood cells originate from the bone marrow and differentiate through hematopoietic stem cells. White blood cells form from granulocytes and agranulocytes. Granulocytes include the neutrophils, eosinophils, and basophils. Neutrophils, eosinophils, and basophils are all forms of a granulocyte cell. When stained, they have granules that are visible inside of the cell. Neutrophils are the most common phagocytic cell. Eosinophils are toxic to parasites and perform some phagocytosis. Basophils are pro-inflammatory cells. Mast cells are similar to basophils and found mostly in tissues and are illustrated here. The group of cells, white blood cells that is, that do not have granules in them when stained are referred to as agranulocytes. These are the natural killer cells, or NK cells, monocytes, and lymphocytes, which are active in adaptive immunity. This is an illustration of the mode of action of a natural killer cell. These are a type of lymphocyte. They can attack cancer and virus-infected cells. Ultimately, their job is to kill cells without normal self-molecules or antigens that identify the cell as belonging to you. Monocytes are the largest white blood cells. In tissue, they become macrophage or dendritic cells and are illustrated here. Pathogen recognition. Most microbes have common chemical patterns. pathogen-associated molecular patterns, or PAMPs. These are very often related to cell wall features. Most cells have toll-like receptors, or TLRs, that attach to them. Upon attachment, cytokines, chemical mediators of immunity, are released that regulate the intensity and duration of the immune response. This is an illustration of that process. Phagocytosis is the ingestion of microbes or particles by a cell performed by phagocytes. Phagocytic cells again include your neutrophils, macrophage, and dendritic cells. This is an illustration of the process of phagocytosis, transcytosis, and eventually exocytosis, where undigested waste is expelled from the cell. Inflammation. Inflammation is the collective response of chemical mediators and cellular defenses to an injury or infection. Acute inflammation is short-lived and localized to the site of injury or infection, while chronic inflammation is inflammation or an inflammatory response which is unsuccessful and may result in the formation of granulomas and scarring. Signs of inflammation include redness, swelling, heat, pain, and altered function. This is an illustration of the inflammatory response. Note each of the aforementioned symptoms. Fever. Fever is an abnormally high body temperature and is a generalized defense used to slow the growth and potentially kill a pathogen simply by elevating the body temperature to a level higher than the foreign pathogen can tolerate. The hypothalamus normally sets our body temperature at 37 degrees centigrade or 98.6 degrees Fahrenheit. Gram-negative endotoxins can cause phagocytes to release interleukin-1 and other cytokines. The hypothalamus can release prostaglandins that reset the hypothalamus to a higher temperature as a result. The body increases its rate of metabolism and shivers, which raise body temperature. At crisis, your fever can break. Vasodilation and sweating can cool the body. Pros and cons of fever. The pros of fever is that it increases transferrins and it increases interleukin-1 activity or inflammation. The cons is that it can lead to tachycardia, acidosis, and dehydration. And this brings us to the end of Chapter 17, the innate, nonspecific host defenses.